and chronic. He was named the 2017 Innovation Fellow by the UNM Rainforest and is also a member of the National Academy of Inventors. Just to save time and keep it easy, I'll also introduce Dr. Catherine Fritz. Catherine Fritz is an assistant professor in the Department of Molecular Genetics and Microbiology. She received her PhD in postdoctoral training at the UNM campus. In 2018, she was selected to be a Clinical and Translational Science Center KL2 Scholar, and an NIH-funded Mentored Career Award has provided funding for her to start her research program. Her laboratory is focused on vaccine development, including using NIH funding to develop vaccines against opioids and also chlamydia trachomatis, which is an ST, a sexually transmitted infection. So we're looking, we've got folks interested in all kinds of stuff. So I'm going to turn this over to Dr. Chakarian, and he can lead us off. Thank you all.
Great. Thank you so much, Kim, for that really nice introduction. Can everybody see my screen okay?
Yes, great, thanks.
Great. Okay. So as Dr. Page mentioned, I'm in the Department of Molecular Genetics and Microbiology, and our lab is interested in developing vaccines for a number of different targets, infectious diseases and chronic diseases. And today, really happy to talk to you a little bit about our efforts to try to develop vaccines for cardiovascular disease. And so as Dr. Page mentioned, our lab, as well as Dr. Fritz's lab, has developed a vaccine platform technology. And we use that platform technology to develop vaccines against a variety of different targets. And this technology is based on something called a virus-like particle. And a virus-like particle is basically what it sounds like. So these are the sort of outer surface proteins or shells of viruses. And it turns out that for many viruses, when you overexpress the structural proteins of those viruses, they form a structure that's basically indistinguishable from the infectious virus, but they're empty shells. So they don't have any nucleic acid inside, so they're absolutely not infectious. And when the virus-like particle technology was first identified in the late 1980s, it became really obvious that those virus-like particles could potentially serve as vaccines for the viruses that they were derived from. And there are a number of examples of VLPs that are used directly as vaccines. And maybe one of the most successful examples of that is the human papillomavirus, the HPV vaccine that was approved in the early 2000s, Gardasil and Cervarix. These are very, very effective vaccines that prevent infection with HPV types that are associated with cancer. But there are many other virus-like particles that are currently in clinical trials. There's a SARS-CoV-2 vaccine, influenza vaccines, and a few other vaccines. And so I think, you know, as these vaccines move through clinical trials, we will see more and more virus-like particles being used clinically as components for vaccines for infectious disease. So one of the reasons that these vaccines are quite appealing is that they are very strong at stimulating the immune system. And so if you look at this virus-like particle from HPV, you see that it has this sort of geometric shape. It's basically one protein that assembles into a pentamer, so 5 copies, and then a number of those pentamers form the virus-like particle. And it turns out that the immune system has developed a number of different mechanisms that allow it to respond very, very strongly to these geometric shapes. I tried to summarize them in this slide, but the details don't really matter that much. But basically, antigens that have these highly dense repetitive structures, when they interact with B cells, and these are the cells that produce antibodies, you get this strong interaction between the antigen, between the virus particle, and the B cell. And the consequences of that is that you get strong signals to the B cell that lead to very, very strong antibody responses. And another really exciting aspect of this technology is that not only do you get these strong antibody responses, but they're very, very durable. And this has been shown with the HPV vaccine, where a single dose of that vaccine can induce antibody responses that stay at a high level for decades after immunization. So they're very, very potent vaccines. So our lab has been interested in basically taking advantage of these properties of virus-like particles, and instead of using them as vaccines to target the virus that they're derived from, we use them as platforms to increase the immunogenicity of other targets and things that are normally poorly immunogenic. So basically the idea is that we start with a virus-like particle, and then we use different techniques where we can array target antigens on the surface of those virus-like particles so that we display that target antigen in the same sort of high density, high valency, highly immunogenic format that the virus-like particle is in. And then we get an antigen displaying VLPs that can induce, again, very strong and long-lasting antibody responses. So we've used this technology to develop vaccines against a number of different things. So we've used this for infectious disease targets. As Dr. Page mentioned, Catherine's lab is using this to target chlamydia. Our lab is targeting malaria and a number of other pathogens. So this is one focus of the lab. But we're also quite interested in trying to develop chronic diseases targeting self molecules that mediate disease. So in this case, basically, we're using the virus-like particles as a way of inducing immune responses against molecules that your body makes, essentially making an autoimmune response, but against molecules that are important in developing disease. And it turns out that one of the nice aspects of this virus-like particle technology is that it's a very efficient way of inducing immune responses against self. Another thing, and this is what Dr. Catherine is going to talk about, is developing vaccines that target opioids. And again, she'll talk about that after I'm done. Okay, so the particular target that I'm going to share with you today is trying to develop vaccines that lower LDL cholesterol. And as I'm sure most people on this call know, elevated LDL cholesterol is one of the most important risk factors for developing cardiovascular disease. So elevated LDL cholesterol is very, very common. About a third of adults in the United States have elevated LDL cholesterol. And statins, which are most commonly prescribed for lowering LDL-C, I think are the most commonly prescribed medications in the US. Lowering LDL cholesterol can be very effective at decreasing cardiovascular events. So here's just some data showing that 20% or a 40 mg per deciliter reduction in LDL cholesterol translates to a 20% decrease in cardiovascular disease events. And even very modest reductions in LDL cholesterol can be very beneficial. And as I mentioned, statins are the most commonly prescribed drug for lowering LDL cholesterol. But statins have their drawbacks. So there are side effects associated with intensive statin therapy, most commonly myalgia. And also, there's a certain percentage of patients who are at high risk of developing cardiovascular disease where statins are not effective. About 20% of patients who have hypercholesteremia. So there's been a lot of efforts in trying to develop other approaches that don't rely on statins to reduce LDL cholesterol. So one of the most exciting targets that was first identified about 20 years ago is a molecule that's called PCSK9. So this is a protein that is secreted in the serum. And PCSK9 has the ability to bind to the LDL receptor. So the LDL receptor is normally expressed on the surface of cells in the liver and hepatocytes. And its role is to take up LDL cholesterol, to internalize it into a cell, and then the LDL gets metabolized by the cell. And then the LDL receptor recycles back onto the surface of the cell, and that process repeats. So PCSK9 acts as a negative inhibitor of this process. So when PCSK9 binds to the LDL receptor, when that receptor gets internalized, instead of recycling back to the surface of the cell, the LDL receptor gets degraded. And so what that means is that individuals who have high levels of PCSK9 have lower levels of the LDL receptor. And when their hepatocytes have low levels of the LDL receptor, that means that they're less able to take up LDL cholesterol, and they have higher levels of circulating LDL cholesterol. So There's very, very strong human genetic data showing that mutations in human PCSK9 can dramatically affect the levels of circulating LDL cholesterol. So what I'm not showing on this slide is that there are actually mutations in PCSK9 that are gain of function mutations. So it's a more active form of PCSK9. And patients who have those gain of function mutations, they have elevated levels of LDL cholesterol and are at much higher risk of developing cardiovascular disease. There are also naturally occurring inactivating mutations in human PCSK9. I'm showing some of the data from some of those studies here. So this is the normal distribution of cholesterol levels in human populations. And in patients who have these inactivating mutations in PCSK9, you see these dramatic reductions in LDL cholesterol levels in these individuals. And it turns out that these individuals who have these mutations in PCSK9 have much lower levels of heart disease than patients who don't have those mutations. These are heterozygous patients who are heterozygous for these mutations. There's a small number of patients who are homozygous for these inactivating mutations, and these patients are absolutely protected from cardiovascular disease. So the fact that mutations in PCSK9 are associated with protection from cardiovascular disease, and also these mutations don't seem to have any side effects at all. These patients are absolutely normal. Suggested that potentially targeting PCSK9 might be an effective way of reducing LDL cholesterol and reducing the incidence of heart disease. So about 5 or 6 years ago, a couple of monoclonal antibodies that target PCSK9 came onto the market. So the trade names of these are Repatha and Praluent. So these are drugs that are incredibly effective at lowering cholesterol levels. And so patients who receive these drugs see reductions of 60% or more, particularly in combination with statins. So they're very, very effective at reducing LDL cholesterol. But one of the problems with these drugs is that they're very, very expensive. So when the monoclonals first came on the market, they were around $14,000 a year. Since then, the drug companies have reduced their prices to around $5,000 to $8,000 a year. But given how common elevated LDL cholesterol is and this expense, these drugs are really only used for patients where statins are not effective at all, or the patients who have side effects associated with statins.
So sorry, I've got another conference on that.
So we've been interested in trying to develop vaccines that target PCSK9 with the idea that a vaccine will require fewer doses. And also the vaccines, because they only need to be used infrequently, less doses, that could potentially increase patient compliance. And there are some other issues with monoclonal antibodies that also a vaccine could potentially address, that I won't go into now. So I just wanted to mention that we're not the only ones who are working on vaccines targeting PCSK9. So this was a very recent study from an Austrian company called Epheris, where they developed a vaccine targeting PCSK9. This vaccine is not particularly immunogenic, partly because it doesn't use the vaccine technology that we're using, but it induces some decent antibody responses against PCSK9. And the vaccine actually was somewhat effective at lowering LDL cholesterol. On average, these patients saw about a 10% reduction in LDL-C. And as I mentioned, even modest reductions in cholesterol can be very effective. And so it's thought that these patients would receive a benefit of being vaccinated. So we're trying to develop a vaccine that can induce stronger and potentially more long-lasting antibody responses against this drug. So what we did was we built vaccines and we looked at the structure of PCSK9. This is it binding to the LDL receptor. We chose a number of regions of that protein, and then we developed vaccines that targeted these different regions of the protein. Basically, the way that we do this is we start with the virus-like particle, and what we have used as a platform is a very simple virus-like particle that's derived from a bacteriophage. So these are viruses that normally infect bacteria, but they don't infect humans. And we use chemistry to link these different peptides on the surface of this virus-like particle so that we display those different antigens at very, very high valency on the surface. Basically, all these red dots on the surface of this virus-like particle can potentially be modified by these peptides. So we get that highly valent display. We chose two different peptides that in preliminary studies were the most effective at lowering cholesterol in mice. And I'm just going to show some of the data from one of those peptides here. So these are mice before we vaccinate them, looking at total cholesterol in those animals. The red dots are the vaccinated animals. The white dots are controls. And you can see before vaccination, these two groups basically have the same levels of cholesterol. But after vaccination, we see a slight increase in the control group. So their cholesterol levels go up as the animals age. But we see this fairly dramatic reduction in cholesterol levels in the animals that received our PCSK9 VLP vaccine. So again, showing proof of principle that this vaccine can effectively work in mice. We were also interested in looking at the longevity of these antibody responses. And so we immunized animals. Again, this was done in mice, and we followed the antibody responses over time. So you can see the antibody responses go up, and then they slowly decline over the course of a year. And so we calculate from this that the antibody half-life is about 20 weeks. And with a half-life of that level, we anticipate that this vaccine potentially would have to be administered every 6 to 8 months in order to keep antibody responses high. But we were most interested in looking at the efficacy of this vaccine in primates. And partly the reason is that mice are really not a great model for looking at cholesterol. They have different lipid compositions. They don't develop heart disease. And so we did a study in collaboration with the Primate Center at UC Davis and looked at macaques that were immunized 3 times with various vaccines or controls. We were able to monitor their LDL cholesterol levels. We wanted to see whether these vaccines worked in combination with statins. And so several weeks after the 3rd immunization, we gave these animals statins. And then we boosted them again before the animals were euthanized. So I just wanted to first show you the antibody responses in monkeys. And so again, I'm not going to go through the details of this, but both of our vaccines induce very strong anti-PCSK9 antibody responses. And then this is sort of the crux of the data, looking at the LDL cholesterol levels in these animals. And so I'm going to take you, this is a lot of data, so I'm going to take you through this one slide at a time. So first, I just wanted to show you the control animals. These just got unmodified VLPs. And you can see over time that their LDL cholesterol levels really didn't change. And that's even when these animals receive statins. And one of the reasons perhaps that these monkeys don't respond to statins is that if you look at their LDL cholesterol levels, they're quite low. So macaques tend to have much lower LDL cholesterol levels than humans. So that's good. So the control vaccine didn't do anything. We next looked at a vaccine where we gave them a single vaccine targeting one portion of PCSK9. And so looking at the early time points, we basically don't see any change. But now when we give these animals statins, so that is these time points here, we see a reduction in LDL cholesterol levels by about 25%. So when we give these animals a single vaccine and then we combine it with statins, we see this effect on LDL cholesterol levels. So the third group we looked at was a group that received a mixture of 2 different VLP-based vaccines targeting 2 regions of PCSK9. And in this group, we saw more immediate effects. So we saw about a 30% reduction in LDL cholesterol levels at the early time points, and that was maintained and didn't go down any further even when we added statins. So this is a vaccine that effectively lowers LDL cholesterol levels in primates, and it does it even without requiring statins. And again, these macaques have low baseline levels of LDL cholesterol. And so we suspect that this vaccine will be more effective when we're targeting a population that has elevated LDL cholesterol levels. We also looked at a number of other markers. And so we looked at ApoB levels. This is the major protein component of LDL cholesterol. And again, the bivalent vaccine worked without statins. The two vaccines work similarly when you added statins. And then we also wanted to look at levels of HDL. So this is good cholesterol, right? We wouldn't expect to see any changes in HDL levels. And we didn't see that, both without statins or with statins. None of the groups have any differences at all. So basically the vaccine is doing what we were hoping that it would do. So just to summarize this work, we've developed these VLP-based vaccines that target this molecule that's critically important in metabolizing LDL cholesterol. And we've shown that we can lower cholesterol levels in multiple animal models and shown that this bivalent vaccine can reduce LDL cholesterol levels in non-human primates, and we can do that in the absence of statins. And so, I mentioned before that we have these monoclonal antibodies that are very effective at lowering cholesterol. And what might be a scenario where a vaccine could potentially be used? And we're particularly interested in these vaccines in areas that have low resources. So elevated LDL cholesterol and heart disease, as I'm sure most of you know, is a huge problem, not just in the developed world, but also in the developing world. And we think that a vaccine-based approach would certainly have advantages, certainly economic advantages, but also potentially practical advantages in low resource settings. And so that's really how we're trying to position this vaccine as we move forward and anticipate clinical trials. So I just wanted to thank a few people. So the people in my lab who are involved in this work. We've been doing this work in collaboration with a group, two groups at the NIH, one led by Alan Ramali, who's at the National Heart, Lung, and Blood Institute, another led by John Schiller at the National Cancer Institute. And our collaborators at the California National Primate Center are also incredibly important in getting those studies done. So I'll stop there. And I don't know if we're doing questions now or if we want to save that till the end.
We'll save questions till the end.
Great.
All right.
Oh, you're muted, Kim.
Oh, and now we have Dr. Fritz.
Thanks. Let me get my share screen up. All right. Can everybody see my screen? Okay, awesome. Okay, so Bryce gave a great introduction to bacteriophage virus-like particles as vaccines. And as he mentioned, I'm going to talk about our work for opioid vaccines today. And this is a collaboration with Bryce. We have funding from the HEAL initiative to do this work, and we have collaborators at some other institutions as well, which I'll mention at the end. So I don't think I have to go over too much background of the opioid epidemic and the public health crisis that we're in. Just to say that, things are getting worse, it seems. And so there's really a call for new approaches and new therapeutics for addressing this crisis. And so you all probably know the current methods. So medications for opioid use disorder, naloxone or Narcan for overdose. But something, of note that I think goes back to what we're trying to work on and why we're working on these vaccines is that current treatments have limitations. So there's limitations in terms of access. Some of these treatments have physical dependence as one of the side effects. There's stigma associated with getting some of these treatments. And then, of course, despite these treatments that we do have, we continue to have overdose rates that are increasing. And so we started doing this work, as Bryce mentioned, we in the past, most of the vaccine work that my lab has done and his lab has done has focused on chronic diseases like heart disease, as Bryce mentioned. We've worked on cancer and then also obviously infectious disease. But our work in opioid vaccines really came about after this call in 2017, which was a review article that was published by the NIH, talking about how science could help the opioid crisis. And so one of those approaches that was mentioned in this review article in 2017 was the possibility for vaccines or monoclonal antibodies as a long-term strategy. And so people had been looking at vaccines for drugs of abuse for a very long time. And there were several groups that had been working on this. And so it was sort of highlighted as a novel approach that could be pursued as a potential solution. So some of you may be asking what an opioid vaccine would even be. So this is just a schematic showing basically the idea of an opioid vaccine. So if you can immunize an individual, probably somebody who's already seeking treatment is the idea. So you could immunize them to elicit an antibody response to a drug of interest. And in this case, it would be opioids. But other individuals have looked at, or other scientists have looked at making vaccines for other drugs of abuse, cocaine, nicotine, and others. If you can elicit these antibody responses, these are proteins that are floating around in your blood, then those circulating antibodies to the drug of interest could bind up the drug in the blood and then prevent that drug from crossing the blood-brain barrier and getting to its receptors in the central nervous system. What this would do is somewhat unclear. So one thing that it's thought would happen is that if somebody takes the drug, then it's not going to elicit the same euphoric effects that it normally would. So you wouldn't have the reinforcement of then the behaviors to go take more drug. So that's the way that it's thought that it would be thought to work for opioid use disorder. We also think that if you can get high enough antibodies in the serum, then there's a chance that you could either prevent or limit overdose. And so that's something that we're really interested in, and I'll talk a little bit more as I go along. As I mentioned, we're not the only ones that are working on this. We initially got involved in looking at opioid vaccines, again, because of this call from the NIH, and that was then translated also into funding, of course. And so we had seen a funding announcement asking specifically for grant applications looking at opioid vaccines. And so we were unable to find anybody who had looked at virus-like particles in this setting. And so we thought it would be worth us trying to do this. So what sort of sets us apart from what other people are doing? Bryce kind of mentioned a little bit about this, but other groups and traditional ways of making vaccines is you take a protein or a peptide of interest, and then you attach it to a carrier protein that can then elicit an appropriate immune response to whatever it's attached to. And so in terms of opioid vaccines, there's a number of different groups. They basically take the drug of interest. In this case, I'm just showing oxycodone. They do some chemistry to attach a linker to that drug, and then they're able to use additional chemistry to attach it to a carrier protein. And these are immunogenic large carrier proteins. Some traditional ones are KLH or keyhole limpet hemocyanin, or tetanus toxoid, which is basically what you get as a tetanus vaccine. These have shown preclinical efficacy. There are some challenges though with how this would be delivered to people. So these primarily these require many immunizations, so sometimes up to 4 immunizations to reach peak titers. It can also the antibody titers, or the amount of antibody that's in the blood after immunization, those can wane very quickly. Bryce mentioned some of this about the durability of responses. And so ideally, we would like like a vaccine that can elicit a very fast response that reaches high titers after, only 1 or 2 doses if possible, and then have those antibodies last a long time. So individuals aren't having to come back and get multiple immunizations over years and years. And so I'm not going to go into too much of the chemistry for this group, but basically we're doing the same approach that Bryce has talked about with the cardiovascular disease vaccines. So we have drugs of interest, so opioid drugs of interest. We have a collaborator at a company called Cell Mosaic, which they are able to attach a linker sequence that we can use to then further attach the drug onto our virus-like particles. And as Bryce mentioned, these virus-like particles, when you attach something to it, there's many, many sites for the drug to attach to. And so we're able to basically try to trick the immune system into thinking the drug is a virus because it's displayed on this virus-like particle that to the immune system just screams, danger. And so we're making the immune system think that the opioid drug is dangerous, which it is. So our overall goal and what we've really been working on right now is just to develop these vaccines and then assess their immunogenicity. So, how quickly their antibody titers are elicited and then also looking at protective responses elicited by these vaccines. And right now so far we've looked at oxycodone, heroin, and fentanyl, and I'm going to show you some of the data from the work that we've done so far. So we started with oxycodone. Oxycodone is pretty easy to get our hands on. And the chemistry that was needed to make the drug suitable for attaching to our virus-like particles was easier for oxycodone. So that's where we started. So we create this oxycodone that has this this short peptide sequence that then allows us to attach it to our virus-like particles. And then we use what's called SDS-PAGE. It's gel electrophoresis to look at our virus-like particle coat protein to see if we've attached that drug to the surface. And so this is just showing that Q beta alone, we see a band here, which is the protein of the expected size for one of those coat proteins of the virus-like particle. And then when we do our fancy chemistry to add the oxycodone, we see a shift in mobility of that band up, meaning that there's more molecular weight on that protein. So we've successfully attached the oxycodone to the virus-like particle, which is what we wanted. Then the first thing that we did was we looked at our antibody responses over time. And so here in this, actually, let me get my little laser pointer. So over here, we have antibody titers. So on the y-axis is how much antibody is in the blood of mice that have been immunized. And then down here on the x-axis is days post-immunization. And so these animals were given one immunization. After 3 days, we took a blood sample and they're already showing antibody responses. This is in the blue line here is our vaccine, our Q beta virus-like particles with oxycodone on it. And those antibody titers can continue to rise up to about 21 days. And then they stay pretty good, go down a little bit, but they're pretty steady over the course of the entire experiment, which we took out to 112 days. I think we actually took it out longer than that. I just don't have that data here. And then just as a comparison, we looked at KLH conjugated to oxy. So this is one of those other traditional protein carriers to see what happens with that. That's one of the approaches that other groups are using for vaccines against opioids. And for that one, we see the antibody titers go up a little bit and then they crash pretty quickly. And then these other lines are controls showing that we don't have antibody responses to those controls as expected. We also wanted to investigate if there was any boosting effect by having additional immunizations. So we gave them a second immunization. This is a separate group of animals, second immunization at 21 days, and then a third immunization at 42 days. And we see that antibodies kind of go up a little bit after a second immunization, but they really stay pretty steady. And a third immunization doesn't really do much. So we think we kind of max out the amount of antibodies that we are eliciting after just a single immunization. And we don't really see much of a difference with males and females. We might see there might be a little bit of a lower amount of antibodies elicited in the males, but it's nothing too striking. So you can have more, and you can have levels of antibody amount that's in the blood, but you can also have different qualities of antibodies. You can have antibodies that bind really strongly to the target, or you can have antibodies that don't bind very strongly. And so we were interested, we hadn't seen much of an effect of additional immunizations on the amount of antibody, but we weren't sure if we were getting a better quality of antibody after multiple immunizations. So we did what's called an avidity index. I won't go into too much detail about that, just to say that as you get higher up on this y-axis, the better the strength of the antibody. And so after one immunization, we see an avidity index around here. These are females, and then males we separated out. And then after a second immunization, we see a stronger antibody binding. So better quality antibodies. And that's not really increased much with a third immunization. We see a similar trend with the males. And so we think really 2 immunizations is probably what we would want, so that we get the highest amount of antibody and also the strongest binding antibodies. So if you remember back to my little schematic of how these vaccines work, what we really are wanting to do is prevent the drug from getting into the brain. And so other groups have looked at retention of drug in the blood in animals that have been immunized as a measure of whether the vaccine is working. And so we did a similar experiment. So we immunized animals, and then we challenged them with oxycodone. And then after, I believe, 15 or 20 minutes, we kill the animals, we decapitate them, and then we collect the trunk blood and we collect the brain. The trunk blood is what I'm showing here, and we measure with mass spectrometry the amount of oxycodone that is in there. And so in animals that have received a control vaccine, just the Q beta virus-like particles, we have a low amount of oxycodone in the blood because it quickly goes into the brain and sort of stays there. In the Q beta oxycodone vaccinated animals, we see increase in the amount of oxycodone that's in the blood, indicating that the antibodies in the immunized animals is retaining that drug in the blood and preventing it from getting into the brain. So this is really good first data showing that the oxycodone vaccine is likely working. So now I want to talk a little bit about our heroin vaccine. So heroin is actually metabolized into 2 active molecules or metabolites called 6-acetylmorphine and morphine. And those are the drugs that we're targeting with the heroin vaccine. And so very similar to what I showed you before, we're able to attach both of those drugs to virus-like particles. And then we went right into looking at immunogenicity, so how much antibody is elicited by these vaccines, and then doing a challenge model to look at actual in vivo drug effect in vaccinated animals. And so we have different vaccine groups. We tested each of the vaccines separately, so Q beta morphine and then Q beta 6-acetylmorphine, and then a combination vaccine that targeted both morphine and 6-acetylmorphine. And so we have mice, both male and female. We do immunizations. Like I described before, we collect blood and look at antibody amounts, and then do a challenge study looking at drug activity. Very similar to what I was showing before. We get antibodies that are high titer, and they last for a long time. This one here is going out to 170 days. And then we also looked at avidity. And so we have a strong antibody binding as well. Very similar to what we saw with the oxycodone. So the challenge model that we used is called a tail flick anti-nociception assay. And so the idea is that if you inject heroin into a mouse, they are going to have, they're not going to feel pain from a painful stimulus. And so the way this works is we give subcutaneous heroin to mice that have been immunized or are control animals. And then we do a tail flick assay. So it is what it sounds like. The animal's tail is, we have a heat source. So it's basically a heat lamp or a laser is a little machine you can buy. It shines the hot light onto the tail. If the animal feels it, they flick their tail away. And you measure the amount of time it takes for them to feel the pain. If they have heroin, you shine the light and they don't really flick their tail away. They will sit there with their tail for a long time because they don't feel the pain. So that time that it takes for the mouse tail to flick is called the latency response. And so this is data from our heroin challenge. So we had animals here. The black lines are Q beta control. Then we have PBS control, Q beta morphine, Q beta 6-acetylmorphine, and then the combined. And you'll see, so on the y-axis, the higher the number, the longer it took them to flick their tail. So they are feeling the effect of the heroin. We would expect that if our vaccine is working, then they're going to feel pain, right? So they're not going to feel the effect of the heroin because the antibodies are binding up the heroin and not letting it get into the brain. So that is what we see. So in the Q beta combined vaccine, the blue line down here, these animals were given heroin and then they feel the pain still because the vaccine is working. And we see kind of we also see reduction as well with Q beta morphine and Q beta 6-acetylmorphine, but it's most striking in the combination vaccine, as we would probably expect. And then this is just showing that data at 30 minutes. And again, we see that Q beta combination vaccine is the best. And so this is showing us that our vaccinated animals, they have antibodies that are blocking the drug activity. Which is really good data for showing that our vaccine is likely working. The other thing that we've done with this is we had a small pilot grant from the UC Davis California National Primate Research Center. We were able to use 3 rhesus macaques per group. So we had a total of 6 animals. We had very simple question. We just wanted to know if our vaccine was immunogenic. So we had 1 and 2 immunizations. We collected blood and then we looked at antibody titers. And this is data from the Q beta oxycodone. So we see in the primates that we not quite as nice as the mouse studies. Of course, mice are, inbred, very easy to work with, and they make pretty good antibody responses. So we see some antibodies being detected after 1 immunization. When we give a boost, it goes up a little bit. And we actually decided to give them a 3rd immunization, and then we see antibodies go up. So we think it's probably likely that, in the mice, it was sufficient to have 2 immunizations. We're probably going to need 3 if we go into humans. But this is without any adjuvant. And so there's lots of ways that we can make this better in the primates and in humans. Finally, I only have really 1 slide on fentanyl, even though it's probably the most exciting one the most exciting part of the project that we have. And we're just kind of getting started on it. So Fentanyl has been a little bit of a challenge for chemistry reasons that I don't think even the chemists really fully understand. But basically fentanyl is a very different compound than oxycodone and morphine. And so it's been more challenging to try to figure out how to synthesize the fentanyl with the linker sequence that we need to attach it to our virus-like particles. But good news is they've tried several different linker sequences, and we finally were able to get something from our collaborators. And it does look like we're getting some conjugation to our virus-like particles. And so that's a great first step for us. And we have these in animals right now. And I have a summer undergraduate who's looking at ELISAs from animals to see if these are actually immunogenic. And so this is really exciting for us. I think fentanyl is something that we're really interested in pursuing, especially because of overdose concerns. We also think that fentanyl, because the amount of fentanyl that people are exposed to, it's such a lower amount that has an effect, that we think it might be more likely that we can really target fentanyl. And so we're really excited to continue doing studies with this. I wanted to just finish up by just talking about some of the future directions. As I mentioned, we're doing studies right now to look at immunogenicity of our Q-beta fentanyl vaccine in mouse models. We're also interested in looking at other alternative endpoints of protection from drug challenge. So I showed you that anti-nociception tail flick assay. That's really just asking, can we block the drug activity in the animals? That has nothing, that doesn't really apply to opioid use disorder. And so there's obviously many more aspects of opioid use disorder and behavioral things that are not being taken into account with animal studies, especially with mice. So moving into more relevant models for that is something that we're interested in. We also are continuing some of those studies that I showed with the blood-brain barrier, retention of the drug in the blood. And we're also really interested in whether our vaccines are able to block opioid-induced respiratory depression. So we're collaborating with Matt Campen to look at that in rats. And so those are sort of future studies that we're going to do. We're also interested in looking at combinations of vaccines. So once we have a fentanyl vaccine, do we want to combine it with heroin vaccines or other combinations? And then just something else to mention as well. I focused on opioids here. I just submitted a pilot grant to the UNM School of Medicine to develop xylazine vaccines. And so that street drug named Trank. So we're going to try to do that. Hopefully we get that funded and we can start some of those studies as well. Other things that we're really interested in, and we don't necessarily have the expertise for, is looking at community acceptability of opioid vaccines. We're in talks with CERC right now at UNM CTSC to sort of think about doing a CTSC pilot that will survey patients to ask about different aspects of delivery of opioid vaccines into communities in New Mexico and what that would look like and what concerns there are, to try to head off clinical trial planning and ideas about, do patients even want this? I think is a question that, has been in my mind for a while. And also, what do physicians and other medical practitioners think about this? That is sort of where we're going with everything. Just wanted to mention our collaborators. So obviously here at UNM, Bryce, and then Matt Campen, and then Isabella Romano, who I believe is on the call as well. She did a lot of this work And this is actually her presentation. And then our collaborators at Duke University, Brandy Johnson Weaver, and then Herman Statz and Bill Wetzel. And then Koen van Rompuy, you've seen this picture. Bryce also collaborated with him. And then our good friend Yume at Cell Mosaic, who has been working on doing the chemistry for us for a lot of this work. And then we have funding from the HEAL initiative and some other pilot awards. So I'm happy to take questions, and I'm sure Bryce can answer questions on all of this as well.
Outstanding. I know this was of great interest to everyone, and we'll take some time for questions. I've got some teed up for you in case we don't get enough. But I would note, Catherine, it's really a good question. You know, why would anyone who is dependent on opioids of any type want to take that? And I think we have to emphasize that this may be more of a harm reduction approach, wherein we save lives by reducing overdose mortality. And but I'd love to hear what other people have to say. And there is one paper, I'll send it to you, about vaccine acceptability, and I can talk a little bit more about that if you want.
Yeah, that would be great.
Lonnie.
Oh,
My inner science nerd is very happy right now. Former biology person. This is really, really cool and super promising. Just Catherine, so that you know, as you were talking about doing some acceptability work, and my group would be happy to collaborate with that. I think I have some folks, and we have access to some populations in Arizona that would be interested in sort of participating in that kind of work. So let's talk later on. One question that kept coming up for me, right? You mentioned it, and it's clear from the chemistry of these molecules that you need a different version of the vaccine, right? For different compounds, you need a different one for heroin versus fentanyl versus oxy and so forth. And the fuckers keep making new drugs. Is there any scenario in which down the road, so how feasible is it to package a bunch of these together? How quickly can you generate a new version and test it and get it on the market when there's a new variant, right? And we've seen this problem coming up a lot recently with COVID. And is there any scenario in which it might be possible to develop a vaccine that targets something that is more of a common feature of these molecules rather than the tails? And I get that the tails are easier to grab, right? But I'm wondering if there's any mechanism by which a vaccine could be developed that might have at least a better shot at generalizing more broadly across the class of drugs, rather than having to have a separate one for every single one that they decide to sell and market.
yeah. So yeah, those are great questions and ideas. So something that we are looking at is cross-reactivity. So do the antibodies listed by the oxycodone vaccine bind to morphine? Those two drugs are closer in their structures. And when we've looked at that, there is some cross-reactivity. And so, but ultimately, I think the experiment we're going to do is immunize with oxycodone vaccine, challenge with heroin, see what happens. And I think that will tell us more. We just don't know right now, sort of what the breadth of our, protection is with that. I don't really expect much cross-reactivity between the fentanyl and the other ones, just because they're so different. But I think that there can be some, so we're limited by the chemistry that's possible. To be able to attach the drug to the virus-like particle. And so, for instance, with fentanyl, I think we had come up with like 4 different versions that we could use. Fentanyl fentanyl and some of the other variants of fentanyl are more similar in structure. And so there may be enough cross-reactivity that it would protect against some of those. But I think the point you're bringing up is absolutely a concern. Regarding how quickly you could make a new formulation, I am not quite sure. Bryce might be able to talk more about sort of like the regulatory aspect of that maybe. And then just the other thing you asked about mixing the vaccines together, definitely we can mix them together. We've already mixed, 6-acetylmorphine and morphine together. And so it's a matter of just making the virus-like particles separately, mixing them together, putting them in at the same time. And we don't really see any effect on the amount of antibody that you're getting when you do that. And Bryce has done some of those studies with other targets.
But from a regulatory standpoint, in terms of like new vaccines, I kind of see this as being analogous to the COVID vaccines, as you've moved from one variant to the next. So I suspect, the regulatory sort of attitude towards, modified versions of fentanyl, for example, would be similar to what we've seen with the mRNA vaccines for COVID. But I'm not absolutely sure of that, but that's kind of my sense.
Well, some of those we already know, there's carbofentanyl out there. There's all kinds of fentanyl has many analogs. And so it's possible we can think about those in advance. But it is a challenge and the market changes all the time. But look how quickly Catherine responded to xylazine. So how about Teresa Williams? I see a hand up for Teresa Williams.
Okay. So I'm going to introduce Bob Phillips. I've got a couple of people sitting here with me for this training. And so Bob Phillips has a question.
Go, Bob.
Good afternoon. Thank you for this work and for this presentation. And my inner science nerd is also quite excited. That's great. I was tracking for some time historically the development of the cocaine vaccine. And I don't know what's actually ever become of that. I think it entered some human trials at one point. But one of the things that I had read some time ago was about some of the acceptability studies. And there was a large proportion of people that would say that they would not want to use it. And I don't know if you've had any similar kinds of expectations around acceptability in our populations. Any correlations with that or thoughts on acceptability across populations like that?
Bob, that's a good question. There's not been a lot of data, there really hasn't been much research on acceptability of opioid vaccines. And I don't recall what happened with the cocaine vaccine. However, first of all, with vaccines, I would say there's always folks who are concerned about vaccines. It's very common. There's many people who are actually very pro-health, but really don't want to take an influenza vaccine. For example, it's for some reason, it's like the one vaccine people really don't want. However, when we work with vaccines and when we're treating and testing them, one thing that's really important to remember is we work with healthy people, right? Basically, we're working with people who are healthy, who are trying to prevent disease. There are some people who would argue that people who use drugs are not healthy, but I would say that most of them are actually pretty healthy. And people worry about things like risk compensation. So this was a huge, huge deal for the HPV vaccine that Dr. Chakarian mentioned, whereby basically people were concerned that if we give folks a vaccine to prevent a sexually transmitted infection, they're just going to go out and have a lot of sex. So it was marketed as an anti-cancer vaccine.
Right.
But I tested a Hep C vaccine, and risk compensation is another thing that we are concerned about. We've studied risk compensation with many, many things, including vaccines. Risk compensation was never seen with HPV. We did not see risk compensation in HIV or hepatitis C vaccine trials with people who use drugs. So the idea that you're just going to go out and become crazier. It may be that it'll happen to one or two people, but for the most part, people integrate this into their prevention quiver. But there will be people who don't like it.
Got it. Hey, thanks.
Julian Trujillo?
Yes, so I'm just for the record, I'm a therapist at a methadone clinic. I'm a LMSW, so I work with a lot of clients that are on Suboxone as well. And with Suboxone, whenever you use any sort of opioid, including fentanyl, there is what we call precipitated withdrawals. What kind of is there a similar effect with precipitated withdrawals with this kind of vaccine in humans that we should be expecting? If so, how severe are they? Because from what I understand, precipitated withdrawals are kind of hellacious.
So I guess the short answer is I don't know. But I think the longer answer is I think the target population for people, I mean, the target population that people are imagining is somebody who has opioid use disorder, is seeking treatment, and this would be an additional option for them. So it wouldn't take over anything else. So I would imagine people would get this vaccine after or while they are already stable on other medication for opioids.
I think that's true, Catherine. And related to that, people do worry about, well, what if you have to have a major operation and you need pain meds? Maybe you could talk about that.
Yeah, so I think that the idea for that is that you would be able to just give them more drug. But there's a possibility. So there's like a threshold, right? Like we can only make so many antibodies. And if you just take more drug than that, then you can just override the response. So it's not it's not going to stop everything. You can definitely override it. And so I would imagine that you would probably take advantage of that if you had to use, fentanyl for a procedure or something.
That's what people do now with buprenorphine. They take more drug. You go in and yeah, it happens, and more methadone. And so we would be looking for therapeutic ways to do that. There was another one. Where did she go? Well, was it Anami?
Hi, yeah, it's Anami. I had a very similar concern / question to what Julian just had. I guess I wanted to also comment that possibly having folks who work at MAT facilities, there is a bit of a power dynamic there where if they offer the vaccine to a client, then the client does feel a bit of pressure just inherently based on the setting. So I was wondering if harm reduction organizations could be a part of the community acceptability research prior.
Yeah.
Fantastic idea.
Yeah, that's a great, that's the sort of thing that we want to understand with the community acceptability is looking at across the board people who interact with patients, and then also individuals who would benefit from the vaccine, and understanding everybody's concerns and understanding points just like you're bringing up of incorporating that information into how a vaccine would then be delivered to individuals. I think beyond using the vaccine for opioid use disorder, I think I mentioned we're more excited about the fentanyl, I think because it could be offered to a lot of individuals, because fentanyl contaminating other drugs, individuals might be interested in having a fentanyl vaccine to help prevent unintended fentanyl exposures. Somebody who's using another drug and doesn't want to take fentanyl or overdose on fentanyl, a vaccine against fentanyl would be really useful. And so that's why we're really interested in, the fentanyl vaccines themselves, but specifically looking at whether we can block opioid induced respiratory depression, which is the main, consequence of overdose for opioids. And so if we can reduce that, then that would be awesome. Because I think you have a whole bigger group of people that can benefit from that.
Thank you so much. I'm sorry that we're out of time. There is a question there about thoughts on methamphetamine. I would also point out to you, Catherine, that xylazine almost always happens in the presence, is almost always nowadays consumed in the presence of fentanyl. And so, you know, you're not barking up two wrong trees there. Methamphetamine, last words.
Yes, we're interested in that too.
Good. Thank everyone for coming. We really appreciate it. Thank you, Dr. Chakarian and Dr. Fritz. We look forward to hearing more about your research and to helping to promote it. I think everyone here is interested, and we will do more work on vaccines and how that works in communities. So we thank everyone for coming, and Christina Phillips, thank you for organizing everything. We really appreciate it. And bye, everyone. Send questions to us anytime.
Please email us if you have questions. Thanks, everyone.